Scientific explanation. Froelich condensates represent a fascinating theoretical concept in which biological macromolecules, such as proteins or membranes, might enter a state of quantum coherence, behaving as a single quantum entity. This idea suggests that under the right conditions, such as energy input from cellular processes, biological molecules could act in unison, similar to particles in Bose-Einstein condensates, which typically occur at ultra-cold temperatures. What makes this concept novel is the suggestion that quantum coherence, traditionally seen in physics under extreme conditions, might also happen in the warm, noisy environment of living organisms. If proven, this could open doors to new understandings of biological processes, potentially improving energy transfer efficiency and coordination in cells. In traditional quantum systems, like superfluid helium, particles condense into a coherent state below a certain temperature. Froelich theorised that biological systems might achieve a similar effect through non-equilibrium energy flow, like metabolism. Analogy. Think of macromolecules in a biological system, like musicians in an orchestra. Normally, each musician molecule plays their part independently. But in a Froelich condensate, they all follow the same conductor, producing one harmonious sound, a single quantum state where all the molecules act in unison.
Let's work through a back-of-the-envelope calculation for Froelich condensates in biological systems based on Froelich's hypothesis. This involves estimating whether quantum coherence could be sustained in biological macromolecules using energy from biological processes like ATP hydrolysis, while accounting for thermal noise in a typical biological environment around room temperature, 300 kelvins. Step 1. Thermal energy in biological systems. First, we need to calculate the thermal energy that causes random motion and decoherence in biological systems. This energy is given by the formula E thermal equals kBT, where Nagion-Naus Kb, Boltzmann constant, is 1.38 times 10 to the negative 23 joules per Kelvin. T is the temperature, around 300 kelvins for biological systems. Substituting values, E thermal equals 1.38 times 10 to the negative 23 joules per Kelvin times 300 kelvins equals 4.14 times 10 to the negative 21 joules. This is the thermal energy associated with random motion at room temperature. Step 2. Energy input from ATP hydrolysis. Now, let's estimate the energy input that drives the non-equilibrium state in biological systems. ATP hydrolysis releases about 30.5 kilojoules per mole of energy. To convert this to joules per molecule, eATP equals 30.5 times 10 to the power of 3 joules per mole divided by 6.022 times 10 to the power of 23 molecules per mole equals 5.07 times 10 to the power of negative 20 joules. This means each ATP hydrolysis event releases about 5.07 times 10 to the power of negative 20 joules. Step 3. Ratio of ATP energy to thermal noise. To see if ATP energy can overcome thermal noise, let's calculate the ratio of ATP hydrolysis energy to thermal noise energy. Ratio equals eATP divided by E thermal equals 5.07 times 10 to the power of negative 20 divided by 4.014 times 10 to the power of negative 21, approximately equals 12.2. The energy from ATP hydrolysis is approximately 12 times larger than the thermal energy at room temperature. This suggests that ATP hydrolysis could provide enough energy to maintain coherent quantum vibrations, at least briefly. Step 4. Coherence time. Estimate. Next, we can estimate the coherence time, T2. The time quantum coherence can survive before decoherence, based on the energy available from ATP hydrolysis. In quantum mechanics, coherence time is inversely related to the energy dissipation, decoherence rate, gamma. Gamma equals 1 divided by T2. Assuming that the decoherence rate is on the order of the thermal energy divided by Planck's constant HH, as a rough approximation, gamma approximately equals E thermal divided by H, where H, Planck's constant, is 6.526 times 10 to the power of negative 34 joules seconds. Substituting E thermal, gamma approximately equals 4.14 times 10 to the power of negative 21, divided by 6.626 times 10 to the power of negative 34, approximately equals 6.25 times 10 to the power of 12 per second. The coherence time T2 is then T2 equals 1 divided by gamma, approximately equals 1 divided by 6.25 times 10 to the power of 12, approximately equals 1.6 times 10 to the power of negative 13 seconds, or 160 femtoseconds. This rough estimate suggests that quantum coherence could last around 160 femtoseconds in a biological system at room temperature, which aligns with timescales seen in other biological quantum processes like photosynthesis. Step 5. Estimating the coherence condition. For Froelich condensates to form, the energy from ATP hydrolysis must not only overcome the thermal noise, but also sustain collective vibrations in macromolecules. The 12:1 energy ratio suggests that ATP hydrolysis could provide enough energy to drive coherent collective behavior, but only for short timescales, around 160 femtoseconds. Conclusion: thermal noise energy at room temperature, 4.14 times 10 to the power of negative 21 joules. Energy from ATP hydrolysis, 5.07 times 10 to the power of negative 20 joules. ATP energy is about 12 times greater than thermal noise, suggesting that quantum coherence could be briefly sustained. Estimated coherence time, around 160 femtoseconds, meaning coherence could survive for very short intervals, allowing processes like synchronized vibrations in biological macromolecules. This back-of-the-envelope calculation suggests that while quantum coherence could exist in biological systems, it would only last for very short timescales, and it would require continuous energy input from cellular processes like ATP hydrolysis to maintain it. This aligns with the core idea of Froelich condensates, where biological macromolecules could enter a coherent quantum state, but only under non-equilibrium conditions with sufficient energy input.
Quantum coherence. In biological systems, is it possible? Quantum coherence refers to the ability of particles to exist in a superposition of states and exhibit collective behaviour over a certain range of time and space. In biological systems, it is challenging to maintain quantum coherence due to thermal noise, but Froelich suggested that under specific conditions, particularly with sufficient energy input, coherence might be achievable in biological macromolecules. Quantum coherence has already been observed in biological systems, such as in the process of photosynthesis, where quantum effects are thought to enhance energy transfer efficiency in light harvesting complexes. Imagine trying to keep a bunch of balloons floating together in the air. Normally, they drift apart, decoherence due to thermal noise. But with the right conditions, perhaps a steady flow of air, energy input, you can keep them moving together in a synchronized pattern. Quantum coherence. The physics behind Froelich condensates. Bose-Einstein analogy. Froelich condensates are conceptually similar to Bose-Einstein condensates, BECs, where particles, bosons, condense into the lowest energy quantum state at extremely low temperatures. In the case of Froelich condensates, the idea is that instead of cooling the system, the energy input from cellular processes like metabolism drives the system into coherence. In a Bose-Einstein condensate, particles move as a single quantum entity, and their behaviour is governed by quantum mechanics. Froelich suggested that, similarly, biological macromolecules might exhibit collective behaviour driven by coherent quantum vibrations. Picture a crowded dance floor where everyone is moving chaotically, thermal energy in biological systems. But if you introduce a strong, steady beat, energy input, the crowd might start dancing in synchrony, moving together as one, similar to how biological molecules might synchronise into a coherent quantum state, energy input and non-equilibrium systems in biology. Biological systems are non-equilibrium systems, meaning they constantly consume and dissipate energy. Froelich hypothesised that these energy flows could be harnessed to generate coherent quantum states in macromolecules. He suggested that energy from ATP hydrolysis, a chemical process that powers many cellular functions, could provide the necessary energy to drive quantum coherence. ATP-driven processes in cells create non-equilibrium conditions, where proteins and membranes are in a state of constant flux. This continuous energy supply might allow certain macromolecules to enter a coherent quantum state, despite the noisy thermal environment. Imagine a coffee cup being stirred. The energy you put into stirring creates organised movement in the coffee, and under the right conditions, the whole liquid can move together in a whirlpool, similar to how energy input could drive molecules into a coherent quantum state in biological systems, biological macromolecules, and Froelich's theory. Biological macromolecules, such as proteins, enzymes, and membranes, are large, complex structures that play essential roles in cellular function. Froelich proposed that these macromolecules could be the candidates for quantum coherence due to their ability to vibrate collectively. These vibrations, if coherent, could lead to macroscopic quantum phenomena in biological systems. In biological systems, tubulin proteins in microtubules, structural components of cells, are thought to exhibit quantum effects, possibly interacting coherently through vibrational modes. Some researchers have speculated that this might play a role in cellular processes such as signal transduction or even consciousness. Think of these macromolecules as a set of pendulums. Normally, they swing at different rates, but if they are linked in just the right way, they could synchronise and swing together in a coherent rhythm. This is what Froelich imagined for biological macromolecules in a quantum state, potential biological functions of Froelich condensates. If Froelich condensates exist in biological systems, they might play a role in processes that require high efficiency and coordination, such as cellular communication, enzyme reactions, or energy transfer. Quantum coherence could enhance the precision and speed of these processes, giving living organisms an evolutionary advantage. In photosynthesis, quantum coherence is thought to help chlorophyll molecules capture and transfer energy more efficiently than classical mechanisms would allow. If similar coherent states exist in other biological processes, they could potentially optimise functions like DNA repair or neural signalling. Think of a factory where all the machines work in perfect synchrony, producing products much faster and with fewer errors than if they worked independently. Froelich condensates might provide similar efficiency boosts in biological systems by ensuring macromolecules operate in unison. Challenges and criticism. Can quantum coherence survive in warm, noisy biological environments? One of the main challenges to Froelich's theory is the thermal noise present in biological systems. Unlike superconductors or Bose-Einstein condensates that occur at near zero temperatures, biological systems operate at warm temperatures, where thermal energy typically disrupts quantum coherence, decoherence. In quantum computing, maintaining coherence requires extreme conditions, such as cooling qubits to temperatures close to absolute zero. In biological systems, maintaining coherence in the warm, dynamic environment of a cell is far more challenging. Imagine trying to keep a group of children silent and focused in a noisy playground. Even though it's possible to get their attention for a brief moment, the chaotic environment makes it difficult to maintain order for long periods. This is the challenge for quantum coherence in biological systems. Experimental evidence and theoretical models for Froelich condensates. So far, direct experimental evidence for Froelich condensates is lacking, though there are theoretical models suggesting how they might arise. Experimental efforts focus on measuring quantum coherence in biological systems under various conditions, often looking for indirect signatures of coherent states, such as specific vibrational modes in macromolecules. Researchers have used techniques like infrared spectroscopy and nuclear magnetic resonance, NMR, to study the vibrational modes of macromolecules, looking for signs of coherent quantum behaviour. However, definitive evidence for Froelich condensates remains elusive. Searching for Froelich condensates in biological systems is like trying to find a hidden radio station in a sea of static. You might pick up faint signals that suggest the station exists, but proving its existence requires more refined tuning and sensitive equipment, Froelich condensates, and the possibility of quantum consciousness. Some speculative theories propose that quantum coherence in biological systems might be involved in higher order processes like consciousness. For example, quantum coherence in microtubules has been suggested as a potential mechanism for consciousness, though this idea is highly controversial and remains unproven. The orchestrated objective reduction, OR theory, proposed by Roger Penrose and Stuart Hameroff, suggests that quantum coherence in microtubules within neurons could play a role in conscious thought processes. While Froelich's original theory didn't extend to consciousness, some have drawn parallels. Imagine the brain as a massive orchestra with billions of neurons acting as musicians. In classical models of consciousness, each neuron plays its part independently, but in quantum models, the idea is that neurons might synchronize at a deeper level, producing a coherent, unified melody that gives rise to consciousness. Froelich. The future of research on Froelich, condensates and quantum biology. Quantum biology is an emerging field, and Froelich condensates are just one of many fascinating ideas being explored. Advances in experimental techniques, such as quantum sensing and ultrafast spectroscopy, could provide new insights into the possible role of quantum coherence in biological systems. Understanding Froelich condensates could open up new avenues for research in areas ranging from medicine to bio-inspired quantum technologies. Future research may explore whether quantum coherence can be harnessed in biomedical applications, such as improving the efficiency of drug delivery systems or enhancing our understanding of neural communication. Just as the discovery of quantum mechanics revolutionised physics and led to technologies like lasers and transistors, understanding quantum coherence in biological systems could revolutionise fields like medicine, neuroscience, and biotechnology, offering new ways to manipulate and control biological processes.